Hi everyone, my name is Ehsaneddin Asgari. I'm a postdoc at Helmholtz Center for Infection Research. Today I'm going to present the joint work of Mufrad Lab at UC Berkeley and McCarty's lab at Helmholtz Center for COF4, where we worked on fine-tuning of language model-based representation for protein functional annotation. And this is a joint work with Andrew Dixon, Maysam Ahmadi, Mohammad Khodabakhsh, Alice McCarty, and Mohammad Mufrad. This project is in the framework of life language processing. Just to briefly introduce life language processing, it started with my PhD at Mufrad Lab at UC Berkeley when I came from an NLP background and we started from proteomics, and then this research got expanded in genomics and metagenomics and also human languages. And then I had the opportunity to expand my dissertation research also with Alice McCarty and Henry Schutze, who are experts in metagenomics and NLP. And since the beginning, the proteomics part of this project is getting expanded in collaboration with these great researchers. I start from some background information about this research and then talk about our approach in COP4 and then introduce our preliminary results, and then I conclude the presentation. Noam Chomsky defines language as a set of sequences constructed from a set of finite symbols. This is a very broad definition of language. By this definition, biological sequences, human languages, and many sequential phenomena in the world, they can be regarded as languages. Like DNA language is a set of sentences out of 4 nucleotides of ATCG. Similarly, RNA and proteins are also sentences out of 20 to 25 amino acids. Existence of a language can be also seen like from the terminologies exist in biology, like transcription and translation, when a DNA is transcribed to RNA and RNA is translated to proteins. Bioinformatics and NLP are natural allies. There have been always methodological exchanges between them. Levenstein distance and Smith-Waterman algorithms, formal languages for expressing biological sequences, they are all examples of these methodological exchanges. In 2015, we introduced language model-based representation for biological sequences, starting a new trend of language processing for bioinformatics, this time using deep learning methods. I start with some basic concepts that we need during this talk. First, language modeling. Language modeling is the task of assigning a probability to a sequence of words. It requires only raw text to be obtained, which means that it's a self-supervised task. And it has information about syntax and semantics of a sentence. So it can distinguish between a good syntax and a bad syntax, and it can verify if the sentence is like semantically correct or incorrect. And it has applications in machine translation, for instance, to distinguish between a good translation and a bad translation, or it can be used in autocompletion. The second concept is representation. We always need to alter data appearance according to the audience that we have. And when we are dealing with a machine, we need numerical values, vectors, and matrices. So in traditional machine learning, much effort was focused on this data representation that required domain knowledge, and it was expensive. Deep learning made automatic representation learning possible. We start from a very naive representation at the input layer, and at the output layer, layer n, we predict the results. The output layer and the input layer are connected through some internal layers, whose parameters are going to be trained based on the error calculated at the output layer, which is backpropagated to the layer 0. Training a neural network may require a huge number of training samples. And if we are working on a general task, the representation that we obtain at the end is also like a general representation that potentially can be applied to other tasks as well. Language modeling is an ideal case for such a scenario. Firstly, we have many training examples for language modeling, as this task is self-supervised. We can just use raw text or raw sequences. And as we discussed, it's a general purpose task, so it has information about syntax and semantics of sentences and sequences. This principle is true for convolutional neural network, recurrent neural network, or like more recent transformer models. One of the specific architectures we focus on in this study is the Skipgram neural net, which is analogous to the language modeling task and proposed by Thomas Mikolov in 2013. In the Skipgram neural network, we try to predict the surrounding of a given word. So given the word, we want to predict the context. And it results in unsupervised training of word embeddings to represent words with a meaningful representation, which contains the contextual information of the words. So words with similar context, they have similar vectors. And it has been shown that this representation is useful in downstream NLP tasks, sentiment classification, part of speech tagging, or parsing, or any kind of NLP task. Let me emphasize that the training of this network is unsupervised, or more accurately, self-supervised. So we only need the raw text or raw sequences to train such a network, and then cut the network in the middle and have the representation for word xt. And when we have enough training samples, we can train a supervised network. So we can just train a classification network using average of embeddings, or using like CNN network or recurrent neural network, or more recent transformer networks. Subsequently, by cutting the network in the beginning, we can have representation for words or subsequences, and by cutting the network at the output layer or close to the output layer, we can have representations for the whole sentence or the whole sequence. And now, what is the main problem that we try to address in live language processing for proteomics? As we all may know, proteins are micromolecules that are main elements in the physiological metabolic pathway of cells. Proteins work as mechanical elements and machineries, so their function is highly tied with their structures as mechanical elements. Protein structures can be described in 4 different levels: the primary sequence, the secondary structure, the tertiary structure, and quaternary structure. The primary sequence determines the 3D structure, however, the mapping function is not known. Problem statement: due to the cost of crystallography and the experiment cost, there exists a large gap between number of known protein sequences and the number of known structures and functions. The large gap between number of known protein sequences in UniProt and the number of known PDB structures shows this large difference. So the question is that, can we use the primary sequence data to improve the protein structure or functional annotation? In the framework of live language processing, we first propose language model-based representation learning of protein sequences to gain prior knowledge from the existing large set of protein sequences and know which sequences have similar context, which can be useful for the low resource scenario we want to predict the function or structure of protein sequences. The second approach we suggest is data-driven protein segmentation, that we mainly use it for motif discovery. And the next, I'm going to briefly introduce the approaches that we use in the live language processing for protein informatics. We introduced the use of Skipgram neural network for training representations for k-mers of protein sequences. That we train this network over a large set of primary sequences, can be SwissProt, UniProt, or UniRep, or any collection of protein sequences. And the trained representations for k-mers can be used for protein informatics tasks. We performed intrinsic evaluation of this representation by investigating the continuity of this representation for biophysical and biochemical properties, and showed that this representation can improve domain identification task as a sequence labeling task, or it can result in accurate classification of protein families or disordered protein sequences. In addition, inspired by this work, k-mer-based representation of natural languages has been also proposed in natural language processing again. So this method got back to NLP again. In the next work, we introduced data-driven segmentation of protein sequences. So the k-mers are not ideal biological words because Ks in the k-mers are usually fixed, but it's not the case in biology. Some important subsequences are large and some are small. However, when we increase the size of K, the k-mer representation can become very sparse. So inspired by byte per encoding method, that is like a text compression algorithm, we proposed a sampling framework of byte per encoding to have multiple views on segmenting protein sequences. This segmentation is data-driven and it divides sequences into subsequences of frequently observed patterns. Just to give an intuition about this approach, we start from sequence of amino acids and then within each step we just merge the top frequent patterns to introduce new symbols. So we go in a space of like larger k-mers but not a fixed k. So we would have variable length segments of protein sequences. And depending on the criteria for stopping of this merging process, we can have multiple views on segmenting the sequence. To find the likelihood of each of the segmentation, we look at the curve of how many sequences are affected in large databases by introducing a merging step, and we draw samples from such distribution to find possible segmentations of a sequence. Then we use this segmentation for discriminative motif discovery. And also we use this segmentation for a variable length learning of embedding space. One finding that we had in this study was that the embeddings, when they are combined or concatenated to the count of k-mers or the subsequences, they improve the result. So the embeddings and the k-mer information, they work as complementary information and improve the underlying functional annotation. As discussed, we use this approach for motif discovery that we evaluated against database of experimentally verified motifs in Elm, and it showed high recall with keeping like a reasonable precision. And more importantly, and more related to kappa, we using this approach by combining k-mers and the embeddings, we got boost in the enzyme, toxin, and subcellular prediction tasks. And with the same approach, we participated in the last kappa pi, and we achieved the first and second places in the biofilm formation prediction. And now kappa 4. In kappa 4, we were given three subtasks. The first task was prediction of gene ontology terms. For the training of the network, we used around 500,000 training sequences from SwissProt that they were annotated for 28K distinct categories, structured in a hierarchical manner. And of course, it's like a multi-label classification task. And the second task was prediction of human phenotype ontology terms that we used around 4,000 training instances that they were annotated for almost 9,000 distinct categories. And again, it was also like a multi-label classification task. And the third task, in which we had lower number of training instances, was prediction of disordered ontology. And we had 1,713 training instances with 45 categories, and it was mainly like a multi-class classification problem setting. As we can see, the second and the third scenarios are low resource setting that we have only few thousand training instances. So this motivates use of transfer learning from a high resource setting like gene ontology prediction or language modeling for these cases. The solution that we suggest is as follows. So we start from the self-supervised or unsupervised training of the language model using Skipgram network that we train over large set of sequences, for instance, 100 million sequences in UniProt. On the next, we use the pre-trained embedding in the Skipgram network in a classification network for prediction of gene ontology. So we use the averaged embedding and we fine-tune this embedding during this training, And by getting the weighted average, we apply a sigmoid layer to predict the gene ontology classes. On the next, when we have like a low resource setting, like this disordered ontology, that we have only 1000 training instances, we fine-tune the representation that is already fine-tuned for the task of gene ontology. So this way we cascade the use of pre-trained embedding and fine-tune them in different levels to achieve higher performance. Brief report on the results. For gene ontology and human phenotype prediction for the purpose of evaluation, we use multi-label accuracy metric that is defined as average of the intersection between the predicted labels and the expected labels over the union. For the gene ontology prediction, we use 382K instances for the training and 154K for the test. And as discussed, we fine-tune the pre-trained skipgram embeddings for this supervised task of gene ontology prediction. And we achieve the multi-label accuracy of 0.3 for 28K labels. For the task of human phenotype prediction, we use 3000 training instances and 1200 cases for the test. First, we fine-tune the skipgram embeddings for this supervised scenario, and we achieve the accuracy of 0.21 for these 45 labels. The second, we fine-tuned the network that is trained for gene ontology prediction in the previous step, and now we fine-tune this already fine-tuned network for gene ontology and skipgram, this time for the human phenotype prediction. And it increased the accuracy from 0.21 to 0.23. Another approach that we included in our submission was prediction of gene ontology terms, this time only for top 100 frequent Go terms in each category of biological processes, cellular component, and molecular function. And we use just multi-layer perceptron as the model, and we test the performance of protein vectors versus concatenation of k-mers with the ProtVec, and we showed that we can achieve high macroresponse when we use the concatenation. And finally, for the evaluation of disorder ontology prediction, which is a multi-class classification task, we use 1,500 training instances and 200 test cases. We report both macro F1 and micro F1. The macro F1 would be more challenging as we have 45 classes and the random chance would be around 0.02. We observed that when we use the fine-tuning of the trained representation, we achieve like a boost in the performance, especially when we have like the cascaded fine-tuning of the representation. So firstly using the self-supervised skipgram and then fine-tune it for the gene ontology prediction. Using this approach, we achieve the micro F1 of 0.86 for top 5 based on the top 5 result, and 0.17 for macro F1 for all classes. And this is also higher, at least in the micro F1, when it is compared with use of ProtVec or concatenation of ProtVec and k-mers using SVM model. Conclusions. Biological sequences can be seen as sentences of the language of life. Language modeling is a general-purpose self-supervised task that can help the downstream protein informatics tasks. Self-supervised and supervised trainings can be cascaded to help a loaded resource scenario of classification. Combination of ProtVec and the k-mer representations can improve the performance. Currently, we are working on different language modeling networks that can be explored for functional annotation. Links to the publications and the code bases for Life Language Processing project can be found on the LLP website, llp.berkeley.edu. Thank you for your attention, and I also would like to thank the organizers for their great effort on making the COPPA challenge and serving the community with this great
shared task. Thank you.